Mendel's first law of inheritance was the law of segregation, and that law Mendel discovered when he looked at one gene at a time. Now, he looked at a lot of different genes, but it was always one gene at a time. So his next question was, well, what happens if I look at two genes or two characters at a time? And he wanted to know, how are those two characters going to be passed from the parents onto the offspring? So what we're now going to talk about are dihybrid crosses. These are called dihybrid crosses because these are crosses that involve two genes at a time, and di does stand for two. So now that we have two genes, we're going to have two different letters, and those letters will represent the two different genes. So if we had a parent that had, say, capital D, homozygous for capital D, and that parent also had an E gene that we were following and was homozygous for capital E, we could take that parent and we could cross it with a parent that is homozygous recessive for both of the genes involved. Now notice, if we're looking at two genes at a time, we've got two alleles of D and two alleles for E in each one of these individuals. This was Mendel's P generation, so this is still the parental generation, and these are going to be true breeding parents because they are homozygous for both of the genes that we're looking at. So Mendel really wasn't too curious as far as what was going to happen when he crossed these two true breeding parents with each other, because if you look at it, there's really only one possibility that we can get. If this parent here is going to undergo meiosis and produce gametes, those gametes are going to be haploid. So those gametes are always going to have one capital D in them, and we know it's always going to be a capital D because that's the only option. As far as the E gene is concerned, we will always have a capital E there. That is the only type of gamete from that parent. The other parent, if it undergoes meiosis, is going to pass on a lowercase d and a lowercase e. No other possibility there. So in this case, when we get our fertilization, we're just taking these two and we're combining them together. Now, when we write these letters, we want to write our D's next to each other, and we want to write our E's next to each other. And the standard way to write these heterozygous genotypes is always to have the capital letter in front, followed by the lowercase letter. So what we're looking at here, this would be the genotype of the F1 generation. And 100% of the F1 offspring are going to be heterozygous for D, and then also heterozygous for E. Now, if you wanted to actually write this into a Punnett square, you could do that. Remember, just as before, we put one parent's gametes on one side, and the other parent's gametes are going to go on the top. And then we just fill in the box based on what is directly across from it and what is above it. And again, we want to group those D's together and we want to group those E's together. So that would be our F1 offspring. Mendel's real question was, what's going to happen in the next generation? So what will happen when this parent right here undergoes meiosis? So how are those eggs going to be formed? How are those gametes going to be formed? So in this case, if we look at how these gametes are going to be formed, we have a little more room on here. If we take our parent, basically what Mendel discovered was that in the gamete, we can get any possible combination of those D's with the E's. So in other words, we could get capital D's with capital E's, we could get capital D's with lowercase e's, and any possible combination of the different D's with the different E's. Well, if we come up with those different combinations, the way that I look at this, there's different approaches you could use. Basically, we could get the capital D with the capital E, so we want to list that here. We could get the capital D with the lowercase e. We could get the lowercase d with the capital E. And we could get the lowercase d with the lowercase e. Now that's one parent, and we would be, if we're trying to produce the F2 generation, we would be crossing two parents like this, so we will be getting the same possible gametes from each of these two parents. Now, as we look down the gametes that we've written here, we've written down 4 gametes that are possible. Notice that every single one of them has one and only one D, and they have one and only one E involved in them. That should be a good way to kind of double check your gametes after you have gotten them written out. Now, another thing to notice is that we can actually look at the genotypes of the parents and predict the number of gametes that we should come up with. That should help us to kind of double check, have we really listed all the possibilities there? So to mathematically predict the number of gametes that we should get from one of these parents, if we look at this parent here, notice that there are two different types of alleles for D. We have a capital D and we have a lowercase d. So we're going to assign that a value of 2 because there's two different ones. As far as the E goes, there's two different E's as well. So we want to assign that a value of 2. Then it's just a matter of multiplying 2 times 2. We do get 4. And that tells us that from this parent, we should have 4 gametes possible if we do go and truly list all of them out. So now if we were going to do the cross, got us a Punnett square here. Now our Punnett square becomes quite a bit bigger because we have 4 gametes from each of these parents. So we're going to take one parent's gametes and list them down the side. So we could say that this is going to be the eggs. And then we write those 4 gametes that we came up with before. And then we take the other parents and we're going to list them across the top. And because it is the same genotype, they are going to be the same gametes as before. So I'm just writing those 4 across the top here. And then it's just a matter of going through and actually filling in all of the boxes. So as we do that, if we go through and we fill in all the boxes, it will take me a little bit of time to get them all filled in for you. Notice that as I go through here, I'm just looking at the box that's directly above and the box that's directly to the side. We want to group the D's together and we want to group the E's together. And if it is a heterozygous combination, we want to always put the capital letter in front. Okay, so as I'm finishing this up, what these boxes are actually representing is all of these in here represent the potential genotypes in our F2 generation. So there are a number of different genotypes inside the boxes here. We could go through and we could assign phenotypes to them as well. When we evaluate the phenotypes for these F2 offspring, we look at the D gene, and as long as they have a capital D, they're going to have that capital D look, whatever it happens to be. If they only have lowercase d's, then they're going to have the lowercase d look. And then we would do the same thing for the e's. Now, if we go through and we list all the possible phenotypes that we could have basically some offspring that have the big D look and that have the big E look to them. So if we count those up, here we would have 1, 2, 3, 4, 5, 6, 7, 8, and 9. Notice that all of those boxes that I crossed off have at least one capital D and at least one capital E. Okay, all of those are going to look exactly the same, even though they do have some different genotypes involved there. Now, if we list another possible phenotype, we could get the big D look combined with the little e look. If we go through and we look at those, now we're looking for at least a capital D combined with 2 lowercase e's. So here we have 1, mark them a little different, 2, and 3. So we've got 3 of those. Another possible look would be lowercase d and capital E. If we count those up, we've got 1, 2, and 3. So we've got 3 of those as well. And then we could also have the 2 lowercase letters here, little d's and little e's. And there's only one here in the bottom box that has that. So if you look at what we've written here, we've written the phenotypic ratio of the F2 generation of a dihybrid cross. This ratio is 9 to 3 to 3 to 1. Notice if you add up all of those numbers, we do get 16, which represents the 16 boxes here in our Punnett square. Now, we could also take a question and we could say, if we take these two parents here, as we did, and we cross them with each other, you could have a question that says, for instance, what is the probability of getting this offspring? Okay, so if we wanted to know just what is the probability of getting an offspring from those heterozygous parents, an offspring that's actually heterozygous for D and heterozygous for E. Well, one approach to that would be really exactly the same thing we did when we looked at the monohybrid crosses, is we do the cross, we draw out the Punnett square, and we count up how many boxes actually have that genotype inside of them. Well, on here, it is actually going to be all of these genotypes that are on the diagonal, at least the way that I've written it here. So there are 4 boxes, and we would put that over the total number of boxes, which is 16, and then that would reduce down to 1/4. So if we wanted to know just when we cross those two parents, what's the probability of getting heterozygous for both of the two genes, the probability is going to be 1/4 or 25%. If we look at this picture here, this is the example that your textbook goes through. Mendel looked at seed traits. So he did have a color gene. It was green versus yellow, and that's represented in this picture with Y's. So we have capital Y representing yellow and lowercase y representing green. And he also had a seed shape, round versus wrinkled. That's represented with R's. So capital R is round, lowercase r is wrinkled. This Punnett square here, you can see the actual phenotypes assigned. So the colors of the seeds are shown and the shape of the seed are shown. Notice a unique thing that happened here compared to what we had in the monohybrid crosses. Now we have some offspring that don't really look like either one of the parents, or they have a new combination of looks to them. So we do have some yellow round, and we have parents that were yellow and round, and we've got the one green wrinkled down here on the bottom. But notice here now that we've got some green round, which we haven't seen before, and we also have some yellow wrinkled, which we hadn't seen before. So when Gregor Mendel looked at the two genes at a time, he came up with this law that he called the law of independent assortment. And what that law of independent assortment states is that basically you can get any combination of the alleles from one gene combining with any combination of the alleles from the second gene when the gametes are formed. So it's really all about the formation of the gametes, which goes back to meiosis, which is what we looked at in chapter 13. If you need to go back and review meiosis because you didn't get it very well the first run through, do go back and take a glance through there and make sure that you do understand how those homologous chromosomes are lining up on the metaphase plate and how they are separating from each other as well. Now this picture here summarizes meiosis for us, and it is definitely a summary, so we're missing a lot of steps in here. But this is the foundation of Mendel's law of independent assortment. So Mendel was looking at two genes at a time, and really we have a pretty big assumption here shown in this blue box when we are talking about the law of independent assortment. So this law applies to genes that are located on different chromosomes. And by different chromosomes, we're talking about non-homologous chromosomes. This is not always going to be the case, for sure. So our chromosomes can have hundreds or thousands of genes located on them, really. So it is possible to have two genes that are located on the same chromosome. But if that is the case, then those genes are not going to abide by this law of independent assortment. That will be something that we look at in chapter 15. As far as chapter 14, we're going to make the assumption that they are located on different chromosomes. So as you can see here, we've got a small chromosome, which happens to have the green gene, or what we're calling A, located on it. And we have a large chromosome, which has the blue gene, which we're calling B, located on it. Now, what we're looking at in this first picture, this would be like a G2 cell. So this would be at the end of interphase after all of the chromosomes have been duplicated or copied. So notice that all of those chromosomes are X-shaped chromosomes, or chromosomes that are composed of 2 chromatids each. Now, as we go through meiosis 1, we're going to be forming the tetrads, and then those tetrads are going to be lining up on the metaphase plate. It is a random lining up of the chromosomes on the metaphase plate, which means that it is possible to get this situation right here, where we get the dark colors, dark green and dark blue, on the same side of the line, and then the light colors on the other side of the line. If that is the case, once those chromosomes are pulled apart and we finish out meiosis, we would expect to get half of our gametes having a capital A, capital B, or the 2 dark colors in them, and we would expect half of them to get the 2 lowercase or light colors inside of them. However, keeping in mind that it is a random lining up on the metaphase plate, it is also possible to get this situation here, where we have, say, light green with dark blue or dark green with light blue. If that's the case, that does affect what we get when we finish out meiosis. So notice here we would expect that half of the gametes are going to have the lowercase a capital B, and half of the gametes are going to have the capital A lowercase b. We don't know how the chromosomes are actually going to line up on that metaphase plate, and it very well may be different every single time we undergo meiosis. So what that means is that we really expect that 25% of our gametes are going to be like this, 25% will be like this, 25% like this one, and then 25% this one. Which means that we're getting an equal probability of getting, say, capital A with capital B, and an equal probability that capital A is going to go with lowercase b. And same thing with lowercase a. Half the time it's going to be with lowercase b, half the time it's going to be with capital B. So that's what we mean by the law of independent assortment. Any combination of the A's is equally probable with any combination of the B's. That is the foundation of our dihybrid crosses. Now, the most important thing, really the most important thing for getting your crosses right when we're talking about the dihybrid crosses, is being able to come up with the gametes. So to come up with the gametes, we need to understand how to basically say capital A with capital B and be able to write that down. Capital A with lowercase b, write it down, and so on. If you can do that, that should help you quite a lot in your crosses, because most students don't really have a problem filling in the boxes of the Punnett square. The difficulty comes in getting the right things on the sides. If you don't get the right stuff on the sides, you're going to have a lot of difficulty getting the right thing inside the boxes. We look at this example. In a certain animal, color, represented by A, and height, which is represented by B, are traits that are controlled by single genes. Green color is dominant to brown color, and tall is dominant to short. Okay, so we want to consider that we have a true breeding short green parent and a true breeding tall brown parent. And then we've got some questions concerning them. If you're dealing with 2 genes at a time, 3 genes at a time, and really if you're dealing with just 1 gene at a time, if you have a question like this, and you're going to have lots of kind of story problems or similar questions on your exam, I would suggest that you go ahead and you write out your different letters that you're dealing with and the phenotypes that they represent. So in this question, we've got a capital A and we've got a lowercase a. So we've got 2 versions of color. We've also got 2 versions of B as well, because we're told that B represents height. So now if we look for color, we are told here that green color is dominant. So that means that we want to give capital A green, and we want to give brown then, by default, is going to get the lowercase a or be recessive. And then if we look at the height gene, we are told that tall is going to be dominant. So that means that capital B is going to be tall and lowercase b is going to be short. Okay, so now if we look at our questions. First off, we want to know what is the phenotype of the short green parent. Keep in mind what the phenotype is. Phenotype is what it looks like. So the phenotype of the short green parent is going to be just that. It is going to be short and it is going to be green. Next question, we want to know what is the genotype of the short green parent. Well, genotype, now we want to use our letters. And notice in the question that we are told that it is a true breeding parent. Remember or recall that true breeding also means homozygous. So it is a homozygous parent. Homozygous for both of the looks involved or genes involved. So if we go back to our letters, it is going to be short, and short is our lowercase b's. So I'm going to put those on here. And then it is also going to be green, which is going to be our capital A's. Got it kind of written underneath there as well. If we look at the next one, the phenotype of the tall brown parent, okay, tall and brown, that's what it looks like. The genotype of the tall brown parent, again, we're going to look at tall. Tall is supposed to be capital B's. We're going to put 2 capital B's because it's true breeding or homozygous. And then we would look for the brown, which is going to be the 2 lowercase a's. So now we know what our parents are. So we had our parents was capital A with lowercase b, and we're crossing that with the other parent. And we want to know what will be the genotype of the F1. Well, to figure out the genotype of the F1, we've got to figure out what gamete are we getting from here. And from that parent, we're always going to get a capital A with a lowercase b. From this parent, we know we're always getting lowercase a because that's the only kind of A we have, and always a capital B. And then we're really just taking the 2 and we're combining them together. So when we put them together, we want to group our A's, group our B's. So our offspring, F1 offspring, are going to be heterozygous for A, heterozygous for B. Then we want to know what are they going to look like. So what will be the phenotype of the F1 offspring from these parents? Well, we look at the A gene. Capital A is green. Okay, so it's going to be green. And then we have the capital B gene, and it's going to be tall. So it is going to be tall and green. 100% of the F1 offspring. So that's a run through of the dihybrid crosses. You do want to look at the dihybrid crosses and make sure that you do understand how to do the dihybrid crosses, because you will have some questions dealing with dihybrid crosses. Now, we just put a blank screen up here. Notice that when we did a dihybrid cross, if we just write this up here, if we were crossing 2 F1 individuals with each other, we said that we had a pretty big Punnett square because we had 2 possible gametes, or 2 possible A's and 2 possible B's. So that gave us 4 possible gametes from that parent. And then this one over here, we had the same thing. So there were 4 possibilities from that parent. And that gave us a Punnett square that actually had 16 squares inside of it, which is pretty large, and it does take a bit of time to fill out that Punnett square. Now, how many genes do you think we inherit at one time? Is it just 1 gene? Is it 2 genes at a time? It's actually thousands of genes at a time that we are inheriting. So looking at it 2 at a time is not really realistic. We can look at it as more than 2 genes at a time. And we will look at it as more than 2 genes at a time. We won't look at thousands, but if we just throw up, say, 3 genes at a time, this would be a trihybrid cross. And when we're looking at a trihybrid cross, notice we've got 3 different letters involved, 1 for each of the 3 genes. So we've got A's, B's, and C's. And notice that we've got 2 copies, or 2 alleles for each one of them. And that's because we're diploid individuals. Okay, so diploid means 2 of each chromosome, which is also going to mean 2 of each allele. Well, if we predict how big that Punnett square would be, we've got 2 different A's, 2 different B's, and 2 different C's. Okay, so 2 times 2 times 2 gives us 8 possibilities. This parent over here, really the exact same thing, 8 possibilities. So notice here, now we've got a square that's got 64 boxes inside of it. Pretty large. So that would take us a while to figure that out. And especially if you consider that that might be 1 question on your test, that might not be something that you want to spend all that time on to actually fill in 64 boxes in the Punnett square. So there are other ways to approach these types of questions rather than draw out the huge Punnett squares. So that will be our next topic that we'll look at, is how to use some statistics and probability rules to avoid these really large Punnett squares. One thing we do want to kind of zero in on and make sure that we do understand here is that we want to be able to take a particular genotype, if we just throw another one up here, and be able to look at it and predict how many different gametes we should be able to get from that parent. So if we look at this one, now we've got a situation where we're not actually heterozygous for every one of the genes involved. Notice here that we're actually looking at 5 genes in this genotype. We've got an A, B, C, D, and E. We try to figure out the number of gametes. Notice we've got 2 different A's, 1 kind of B here, 2 different C's, 2 different D's, 2 different E's. So when we go through and we multiply this, 2 times 1 is going to give us 2, times 2 is 4, 8, and then 16. So from that parent, we have 16 possible gametes. Want to just emphasize here, make sure that we are multiplying by the 1 and not adding the 1. That is a very common math mistake that students make on these. So 16 different possible gametes from that parent. If we were just to list a few of them, just to make sure that we do understand what those gametes would look like, they would be something like this. Any possible combination of the A's, B's, C's, D's, and E's that we have. And make sure when you look at your gametes that you've got 1 and only 1 of each of the genes that we're actually looking at here.